A deeper look into signal transduction, G-protein receptors, and cyclic AMP. Signal transduction consists of reception, transduction, and response. First, ligands, such as ions or molecules from the external atmosphere, attach to receptors embedded in the cell membrane. The ligand-receptor relationship works like a key in a lock. The ligand binding site on a receptor is a specific shape, and therefore ligands must fit that exact shape in order to attach. The receptors, specifically metabotropic receptors, are then stimulated to send a signal to heterotrimeric G proteins. Heterotrimeric G proteins are made of alpha subunits and beta and gamma subunits. The G protein starts off binded with a GDP, or guanosine diphosphate molecule, which inhibits the protein's activeness. This is also known as protein conformation, since the attachment of the receptor changes the shape of the protein and its ability to function. This GDP is attached to the alpha subunit as well as the beta and gamma subunits to keep the G protein inactive. When a signal is sent from a receptor to a G protein, the phosphorylation, the addition of a phosphate, of GDP occurs and the beta and gamma subunits disconnect to create GTP, or guanosine triphosphate. This allows for that G protein to be stimulated and pass on the signal from itself onto the next protein in the signal transduction cascade. Once the signal has been sent from that G protein, dephosphorylation, the removal of a phosphate of GTP to GDP, occurs and the beta and gamma subunits bind to the alpha subunit to inactivate that G protein for later use. The repetition of the cycle continues until the signal reaches a final protein that distributes a cellular response to regulate whatever cellular activity the original signal was meant for. If we go back to the beginning, we can see the synthesis of second messengers takes place. The purpose of these messengers is to initiate and coordinate the present signal transduction. The enzyme adenylyl cyclase, found in the cell membrane, activates when GTP binds to the G protein alpha subunit and converts ATP to CAMP. Next, the CAMP activates the enzyme protein kinase A, which phosphorylates GDP to GTP. This then activates the G protein to transfer the signal onto the next protein. The protein kinase A is very dependent on the amount of CAMP. When the signal is finished passing through the activated G protein, the enzyme phosphodiesterase breaks the phosphodiester bond in CAMP to inhibit its work, which leads to the deactivation of that G protein. Once all these steps have occurred, the cell has now received its signal in an efficient manner and can perform work in the cell to contribute to the overall function of the system of the body.